Good afternoon. My name is Alyssa Baker, and I will be presenting here today on my master's thesis titled The Historical Biogeography of Spiny Lobsters in the genus Panoris and the Influence of Environmental Conditions on the Adaptive Evolution of Mitochondrial Protein-Coding Genes in the Aculeata. About 71% of the Earth's surface is covered in ocean, making marine ecosystems some of the largest ecosystem types on the planet. Current marine research is concerned with factors that affect biodiversity, including resource harvesting, pollution, changes in geography, and fluctuation in habitat parameters. Biodiversity is influenced by both contemporary and historical changes in environmental conditions that are most commonly mediated by geographic changes over deep time, such as through shifting tectonic plates. So, why do we care about marine biodiversity? Well, biodiversity promotes the ability of an ecosystem to resist change and provides ecosystem services such as oxygen and nutrient cycling, fishery harvests, jobs, and leisure activities. If biodiversity is to be maintained, management policies must accurately account for species responses to both future geographic events and major human-mediated environmental changes. Diversification describes the increase in distinct lineages within a clade over time and is more specifically defined as the rate of speciation minus the rate of extinction. Oftentimes, divergence in marine systems is primarily facilitated or constrained by highly dispersive planktonic larvae, which can allow for population connectivity over broad ranges. However, this connectivity is also influenced by geographic changes that interrupt or alter dispersal patterns for significant periods of time. As species and populations disperse, they colonize habitats with variable environmental conditions and thus different selective pressures. The infraorder Aculeata is one such model system with highly dispersive larvae influenced by geographic changes. The Aculeata consists of the spiny and slipper lobsters, which are characterized by a long-lived planktonic phylosomal larval stage and a lack of claws on the first set of legs or periopods. Their diverse morphology and global distribution across a wide range of environmental conditions also makes the Aculeata an ideal system for studying historical changes in both diversification and selective pressure. Within the Aculeata are two families, the Palynuridae, consisting of spiny lobsters with elongated antennae and sharp carapace spines, and the Celaridae or slipper lobsters, which are dorsoventrally flattened and possess enlarged paddle-shaped antennae. Within the Palynuridae are two groupings, the Stridentes, which possess a stridulating organ for sound production, which I have highlighted here in blue, and the Silentes, which lack this organ for sound production. The largest genus of spiny lobster, and also the largest genus in the Stridentes grouping, is Panoris. Panoris is also the most economically important group, with fisheries valued in the billions of US dollars. My overarching thesis goal was to clarify the historical biogeography of Panoris spiny lobsters, as well as reveal the geographic changes and selective pressures that contributed to their prior divergence and modern distributions. So as such, my second chapter aimed to test the historical biogeography of Panoris and reveal plausible conditions that affected divergence over geographic time, and my third chapter aimed to evaluate how environmental conditions drives the adaptive evolution of mitochondrial protein-coding genes, or PCGs, in the Aculeata. I'll begin by discussing the historical biogeography of the genus Panoris in my second chapter. Historical biogeography informs on community assembly and species richness patterns. It reveals answers to questions involving the original location of ancestral populations and the specific scenarios surrounding how and why divergence occurred. Population divergence and dispersal is modified by factors such as extinction, geographic isolation, also called vicariance, as well as by the colonization of new habitats, and it is heavily affected by the dispersal ability of an organism. Marine systems are primarily impacted by two factors: plate tectonics or continental drift, and ocean currents. Plate tectonics prompt environmental changes that contribute to further diversification. Examples include mountain uplift, volcanic activity, the formation of mid-ocean ridges or trenches, a new emerging coastal habitat, and the opening or closure of seaways. The opening or closure of seaways and creation of new land barriers via plate tectonics additionally affects current patterns, including their path of travel, strength, and upwelling. Changes to current patterns can in turn affect regional or global climate and sea levels. Currents also have a direct role in facilitating or constraining the dispersal of long-lived larvae, because shifting plates often change the pattern of specific currents used for dispersal, which can in turn either promote population connectivity or result in vicariance events that limit dispersal. Prior inferences proposed by George and Main for the historical biogeography of the Aculeata suggested that vicariance caused by changes resulting from continental drift was the main mechanism behind pinnar speciation from the Paleocene to the Pleistocene. Divergence was expected to have been influenced by mountain uplift and runoff during the Paleocene and the Eocene. Oftentimes, additional mountain runoff creates coastal regions with lowered salinity and higher turbidity. During the Miocene, divergence was expected to have been influenced by changes to upwelling and current patterns in the Indo-Pacific, where numerous ancestral species were expected to be residing. And the Pliocene saw the uplift of New Guinea and the emergence of new coastal habitat, such as the island chains around Easter Island. And lastly, the Pleistocene was expected to see periods of isolation caused by glaciation. My goal for Chapter 2 was to test and thoroughly describe the historical biogeography scenario proposed by George for pinnar spiny lobsters. Pinnars is an especially interesting genus because of their convoluted evolutionary relationships that are not readily defined by modern-day geographic boundaries. I initially proposed that an ancestral state reconstruction would confirm the divergence times, locations, and rates suggested in the comprehensive historical biogeographic scenario proposed by George. Although I tested 4 of the hypotheses proposed by George, I'll describe the 3 most important ones here. So first, it was hypothesized that the ancestral species of the Stridentes and the Silentes originated in the southwestern Pacific during the late Jurassic about 145 million years ago. I also anticipated that the ancestor of Pinnar lineages 1 and 2 originated in the eastern Pacific and southwest Atlantic during the Miocene, 23 million years ago to 5 million years ago. Lastly, most ancestors of Pinnar's extant taxa were expected to emerge in the Indo-West Pacific during the Miocene, about 9 million years ago, with diversification continuing until the early Pleistocene, about 1 million years ago. To address these hypotheses, I constructed maximum likelihood and Bayesian inference phylogenies based on morphology and gene fragments, then created a time-calibrated tree using fossils, and reconstructed ancestral ranges and compared these to a time frame of known geographic events affecting marine systems. I first built the morphological and gene fragment data matrices using 59 members of the Aculotta. Specifically, my in-group consisted of 25 species and subspecies of Pinnar, as well as 24 other species representing all other genuses in the Palynuridae. The out-group consisted of 10 members of the Siluridae family. Morphological data was sourced from holotypes and species descriptions, as well as from physical specimens when available. To build the morphological dataset, physical characteristics were assigned a number corresponding to specific features and a key. 83 characteristics were assessed per species, and any uncertain characters were left blank. Some shortcomings of morphological data include the chance of bias in which characteristics are accounted for in the assessments, as well as for the potential of convergent trait evolution. However, considering the limited available molecular data for some species, by evaluating both morphological and gene data, the overall assessment is more comprehensive. To build the genetic dataset, 180 sequences were sourced from GenBank, and 70 sequences were generated from 19 laboratory specimens representing 15 species in the genus Pinnar. 5 gene fragments were utilized, 2 of which were mitochondrial and 3 of which were nuclear. So phylogenetic relationships were first reconstructed. The morphological dataset was compiled from the key. The gene fragment dataset was aligned in MEGA with gaps removed in G blocks, and the most suitable model selected using J-model test. For both the morphological and gene fragment datasets, maximum likelihood trees were generated in IQ tree, and Bayesian inference trees were generated using Mr. Baez. So for the phylogenies produced using morphology, both the maximum likelihood and Bayesian inference trees had poor support across most nodes, so we'll be focusing on the trees produced from gene fragments. On the trees produced from gene fragments, within the Aculotta, there was full support for the monophyletic Palynuridae and Siluridae families. Within the Palynuridae, there was full to strong support for a Silentes clade and moderate to poor support for a Stridentes clade. The Silentes clade contained the Palynuridae species with the earliest divergence, Philamphion elegans, formerly known as Palinurellus wynecki. Within the Stridentes clade, there was full support for a monophyletic Panoris clade containing the fully supported sister lineages 1 and 2, with lineage 1 tending to be species that prefer clear coastal habitat, and lineage 2 containing species that tend to prefer turbid shallow regions influenced by runoff. The species with the earliest divergence in Panoris was Panoris interruptus, which contradicts prior reconstructions, including Patek and Oakley 2003, which George 2006 had utilized to help inform numerous hypotheses. Numerous prior trees, including Patek and Oakley 2003, had suggested instead that Panoris argus had the earliest divergence. I would also like to highlight that no distinct Panoris longipace lineage was recovered, and I suggest the subspecies Panoris L. longipace and Panoris L. bispinosus should be raised to the species level. Both maximum likelihood and Bayesian inference, gene tree topology, and branch lengths go against the currently accepted nomenclature, which would be expected to confirm a distinct P. longipace lineage with a sister relationship between the subspecies. However, instead, the phylogenetic analyses provide evidence supporting a sister relationship between Panoris femorostriga and Panoris L. bispinosus. Additionally, Panoris homaris was recovered as the most recently diverged species in Panoris. Currently accepted nomenclature considers Panoris H. homaris and Panoris H. megasculpta the same subspecies Panoris H. homaris, which this study supports. Additionally, there was support for raising Panoris H. rubellus to the species level. Both maximum likelihood and Bayesian inference tree topology recovered branch lengths for Panoris H. rubellus that were comparable to the degree of evolutionary difference between other species within the genus Panoris and within other species of the Aculotta. However, a population genetic study conducted by Laverly et al. 2014 indicated that although Panoris H. rubellus was genetically distinct, it is not fully reproductively isolated from the other Panoris homaris subspecies. To time calibrate the phylogeny based on gene fragments, 4 fossils were assigned to nodes. Each of these fossils were confidently identifiable to the species level and formed in stratigraphic levels that had relatively narrow age estimations. The fossilized species of Uinopalinaris scrami was assigned to the node representing the Aculotta. Silardes punctatus was assigned to the node representing the Silaridae. Archaeopalinaris levis was assigned to the node representing the Palinuridae, and Panoris de Stumbasi was assigned to the node representing Panoris. The file to configure the analysis parameters was first constructed using the GUI package Beauty, with probable node ages throughout the phylogeny, then being calculated using BEAST, which estimates probable divergence times from the posterior distributions of the trees. And the resulting divergence time estimation reconstruction, each node represents median ages, and at each node is a 95% highest posterior density interval, or HPD bar, that represents the approximate potential age range for each node. During the Triassic epoch, the Aculotta emerged about 230 million years ago. The Palinuridae diverged about 221 million years ago, and the Stridentes diverged about 210 million years ago. During the Jurassic epoch, the Silentes diverged about 179 million years ago. The Silaridae family diverged about 171 million years ago, and Panoris diverged about 161 million years ago. During the Cretaceous epoch, lineage 1 emerged about 133 million years ago, and lineage 2 diverged much later at about 85 million years ago. Most ancestral species in Panoris diverged in the Eocene epoch, with divergence continuing until the Pliocene. And I would, again, like to highlight that Panoris L. longipace and Panoris L. bispinosus had drastically different divergence times. Additionally, Panoris H. rubellus had a significantly earlier divergence time than the other Panoris homaris subspecies. This difference was about 25 million years with no overlap between the 95% HPD bars. So next, ancestral states were reconstructed. Up to 6 potential geographic areas were allowed as potential ranges for nodes across the phylogeny. Use of any more than 6 possible areas was too time-intensive to run with 59 taxa, but it would be interesting to conduct reconstructions for subsets of these areas in the future. Extant taxa were assigned a combination of these ranges that best matched their present-day distributions. The time-calibrated estimation of ancestral ranges over geological time were reconstructed using 2 methods in RASP. First, the Bayesian binary method was executed with fixed Jukes Cantor or estimated F81 state frequencies with gamma among site variation. Bayesian binary method more frequently suggests single distribution areas for nodes, which is why many historical biogeographic studies still execute this method today. However, Bayesian binary method is more simplistic than BioGeoBears, and it copies ancestral ranges to both possible descendants with 100% probability, which is unlikely for most scenarios. Additionally, the ancestral ranges recovered using the Bayesian binary method were not very well supported, so I'll focus on the second analysis of BioGeoBears. BioGeoBears uses Bayesian's time-stratified reconstructions that consider a combination of free rate dispersal, bicariance with and without founder event speciation, which is where an ancestral species colonizes a new emergent habitat, and it also considers extinction events. BioGeoBears can test 3 possible models, DEC, Diva-like, and Bay Area-like, with or without the J parameter that represents founder event speciation. The most suitable model was determined using the BioGeoBears model test, which evaluates data via a likelihood ratio test, or LRT, and a nested Akaike information criterion test, or AIC. LRT compares the fit of 2 nested models, so a model with versus a model without J. So for example, DEC versus DEC plus J. And it uses p-values to reject the null that models with and without J are equally likely to fit the data. Meanwhile, AIC compares both the nested and the non-nested models. However, which model has the best fit is only relative to other models included in the test. Bay Area-like plus J was found to represent the most plausible reconstruction of Penore's ancestral ranges, and Bay Area-like plus J allows ancestors to inhabit multiple areas, and it allows for founder event speciation, but not a subset sympatry nor subset bicariance. The resulting ancestral range reconstruction showed that the Aculeata emerged about 230 million years ago during the Triassic epoch and dispersed throughout the warm Paleotethys and Tethys oceans during a period of extensive volcanic activity. The Palinuridae diverged shortly after, about 221 million years ago, and likely experienced similar conditions in the Tethys Sea. Within the Palinuridae, the Stridentes diverged in the Western and Indo-Pacific about 210 million years ago during the late Triassic, and the Silentes diverged in the Indo-Pacific about 179 million years ago during the Jurassic epoch. The Stridentes and Silentes are likely influenced by the unification of the Tethys Ocean with the Paleotethys, as well as by the formation of the Tethyan Trench along the southeastern Laurasian coast. Within the Stridentes, the common ancestor of Penoris emerged in the West and Indo-Pacific about 161 million years ago during the Jurassic epoch. By this time, the Tethys Ocean had unified and the Tethyan Trench had begun to form. The lineage 1 ancestral species and the lineage 2 ancestral species originated in the Western and Indo-Pacific during the Cretaceous epoch. This period was characterized by drastic northern movements of the African, Indian, and Australian continents that narrowed the northern Tethys, expanded the Indo-Pacific, created mid-ocean ridge formations, and introduced new shallow turbid regions. The greatest concentration of Penoris ancestral species existed in the Indo-West Pacific from the Eocene, about 52 million years ago, to the Pliocene, about 3 million years ago, in response to the heightened presence of dispersal and vicariance events. These namely included the opening and closure of seaways, current fragmentation and formation, and fluctuations in environmental conditions such as global cooling and the emergence of new turbid low salinity coastal regions. To summarize, the hypotheses proposed by George and others were partially supported. My study supported dispersal and secondarily vicariance caused by shifts in plate tectonics and current patterns as the main driving forces behind speciation within Panoris. This is in opposition to George's assumption that vicariance mainly drove Panoris diversification. Although generally most ancestral species within Panoris diverged earlier in the model than the epochs proposed by George and others, several divergence times for ancestral nodes within Panoris did agree with the suggested epochs and types of geographic changes. Geographic events likely significantly impacted the dispersal and settlement of long-lived phylosoma larvae, thus altering gene flow patterns. Key divergences included the dispersal of the Aculeata throughout the Paleotethys and Tethys about 230 million years ago, the divergence of Palinuridae in the Tethys about 221 million years ago, the divergence of Stridentes in the Western Indo-Pacific 210 million years ago, and the origin of the ancestral species of Panoris in the Tethys Ocean 161 million years ago. Future considerations include taxonomic revisions to Panoris L. longipes, Panoris L. bispinosus, and Panoris H. rubellus. Additionally, one should consider potential flaws in morphology-based trees. Groups with many species in regions of similar selective pressures might produce trees that incorrectly assume relationships due to convergent traits. Future studies should consider further clarifying the validity of these subspecies through population genetic studies. Additionally, the discovery of any new fossil that can be confidently described and dated can drastically affect historical biogeography modeling. So to produce the most accurate inference, it's recommended that the divergence times in the ancestral areas are modeled again as new fossil information becomes available. It is important to consider that broad-scale geographic changes also alter regional environmental parameters. Therefore, as Panoris species and populations diverged in response to geographic change over time, they colonized habitats with variable environmental conditions and different selective pressures. The evolutionary effect of these environmental changes on extant taxa will be further explored in my third chapter. Adaptive evolution occurs under prevailing biotic and abiotic environmental conditions and changes the prevalence of alleles in population gene pools. Mitochondrial genomes experience a greater mutation rate than nuclear genomes, possibly due to exposure to oxidation damage in the mitochondria, and because maternal inheritance lacks recombination that would otherwise help counter mutations. As such, mitochondrial protein-coding genes are susceptible to site-specific substitutions that might alter protein structure or components. Signatures of adaptive evolution on a gene can be identified by evaluating the ratio of Ka to Ks, where Ka represents non-synonymous substitutions per non-synonymous sites, where there's a base substitution that changes the amino acid sequence being produced, and Ks represents synonymous substitutions per synonymous sites, where there's a substitution of bases that does not change the amino acid. Oftentimes, the ratio of Ka to Ks is represented by omega. So negative selection occurs when there are decreased non-synonymous changes, so more synonymous substitutions than non-synonymous substitutions, which suggests that amino acid diversity is potentially constrained and that deleterious mutations might be removed. Neutral selection occurs when the ratio of non-synonymous to synonymous mutations is the same, which potentially suggests that mutations are accumulated fairly randomly, or that equal parts of the gene are under positive or negative selection. And lastly, positive selection is when multiple non-synonymous substitutions are favored, so there's more non-synonymous substitutions than synonymous, and thus it's likely that increased amino acid diversity is favored. The 13 mitochondrial protein-coding genes encode for proteins utilized in the OXPHOS pathway, which is vital for energy production within cells. The first four complexes form the electron transport chain, which builds a proton gradient in the intermembrane space. Complex 1, NADH dehydrogenase, is where protons enter and bind to NADH. Complex 2, succinate dehydrogenase, is the only complex primarily encoded on the nuclear genome, so I won't go into detail here. Complex 3, cytochrome C octoreductase, oxidizes ubiquinone to ubiquinol. And complex 4, cytochrome C oxidase, binds electrons to oxygen to form water. The final complex, complex 5, is ATP synthase, and this is where most ATP is produced via the oxidative phosphorylation of ADP. It is suggested by numerous prior studies on the adaptive evolution of mitochondrial protein-coding genes that complex 1 and 5 are influenced by environmental factors. Complex 1 is responsible for forming a proton gradient by oxidizing NADH, then pumping hydrogen ions from the matrix to the intermembrane space. It is thought that temperature potentially impacts oxidation and pump speed. In ectotherms, which cannot regulate body temperature, the environmental temperature may fluctuate outside of the range for optimal enzyme function. And exceeding the optimal temperature range has been shown to increase mitochondrial activity and also oxygen consumption. When oxygen consumption is greater than what the circulatory system can deliver, then cellular hypoxia results. And this in turn prompts elevated respiration in order to avoid whole body decline. Therefore, it is likely that ectotherm complex 1 sensitivity reflects environmental temperature fluctuations, with more narrow sensitivity ranges in tropical species and more tolerant ranges in temperate species. Complex 5 takes hydrogen ions from the intermembrane space and drives these into the ATP synthase enzyme in order to perform oxidative phosphorylation on ADP and thus produce energy in the form of ATP. ATP production is thought to respond to cell demand, with increased ATP demand and therefore elevated proton channel activity occurring in species that inhabit perpetually low oxygen conditions, such as in marine crustaceans inhabiting hydrothermal vents, as well as in deep water corals and numerous high altitude species. In order to maximize the efficiency of this complex, there are therefore likely structural differences specific to dissolved oxygen concentration at the specific inhabited depth. I aim to first build upon genomic resources by assembling and describing the complete mitogenomes of Panoris echinatus and Panoris interruptus. I then evaluated the influence of consistent versus fluctuating temperatures and low versus high oxygen environmental conditions on the adaptive evolution of mitochondrial protein-coding genes in the aculeata. So why use the aculeata to examine the adaptive evolution of mitochondrial protein-coding genes driven by differences in temperature fluctuations and oxygen concentration? Well, the aculeata are a fairly large, diverse group of about 150 species with a global distribution spanning a wide range of latitudes and depths. They also experience extensive differences in habitat and environmental conditions, and since these environmental conditions should affect oxphos efficiency, it is possible to test for adaptive evolution or positive selection on the protein-coding genes of interest. The Paloneuridae inhabit tropical and subtropical regions, with warm tropical regions tending to have the most consistent temperatures. Meanwhile, the Celaridae inhabit tropical, subtropical, and temperate regions, with temperate regions tending to have the greatest temperature fluctuations. I therefore expected that Panoris spiny lobsters inhabiting equatorial tropical conditions with consistently high temperatures would experience stronger positive selection on NAD2, 4, and 6 because complex 1s were refined to operate within an optimal temperature range, and tropical regions tend to have the narrowest temperature fluctuations. The aculeata also inhabit a wide range of depths. The Paloneuridae typically inhabit 0 to 1,300 meter depths. Meanwhile, the Celaridae typically inhabit 1 to 850 meter depths. Although it looks like at a glance the Paloneuridae inhabit the deepest depths, in actuality, with the exception of the genus Porulus, most paloneurids prefer shallower regions. For instance, one genus of interest, Panoris, usually inhabits 0 to 100 meters depths, with the occasional 180 meter exception. A larger percentage of the Celaridae, however, are deep water species. Most of the Celaridae species for which I have genomic data have depth ranges that extend into 200 meters. However, dissolved oxygen concentration is substantially greater towards the ocean surface than at these depths exceeding 850 meters. I thus hypothesized that Celaridae with known depth ranges over 200 meters would experience positive selection on ATP 6 and 8 because comparatively low oxygen waters at depth will require elevated complex 5 activity to maintain the proton gradient. To address these hypotheses, I first assembled new genomic resources, and then constructed a maximum likelihood phylogeny using 13 mitochondrial protein-coding genes for all available aculotta with complete mitogenomes. And I then tested for adaptive evolution on mitochondrial genomes across this phylogeny. Before evaluating selective pressure across the aculotta, I expanded upon available genomic resources. The complete mitogenomes of Penorus echinatus and Penorus interruptus were sequenced from periopod or walking leg muscle tissue using Illumina paired-end shotgun sequencing, and then was assembled via the Git organelle pipeline. I first annotated and described the mitogenomes, then conducted numerous analyses on composition, specifically nucleotide composition and codon usage. I also analyzed various other components, such as protein coding gene selective constraints, tRNA secondary structure, and control region structure. Both resulting mitogenomes were about 15,600 base pairs long and contained 13 protein coding genes, 22 transfer RNA genes, 2 rRNA genes, and a non-coding control region thought to help regulate gene expression. Additionally, both mitogenomes had a slight AT codon bias. Additional details on codons, tRNA secondary structure, and control region info were also calculated, and if you're interested in these components, I would like to invite you to visit one of my poster sessions during an upcoming Biosci Research Expo. However, I want to focus primarily on discussing selective pressure during this presentation. Next, I constructed a maximum likelihood phylogeny using all 13 protein coding genes. This phylogeny included 34 species, namely 12 species in the genus Penorus, 15 additional species from the family Palynuridae, 7 species in the Siluridae, and an outgroup consisting of Alphaeus japonicus and Nephrops norvegicus. Alphaeus is in the infraorder Caridae, which consists of the shrimps, and Nephrops is in the infraorder Astacidae, which consists of clawed lobsters and crayfish. Sequence datasets for each protein coding gene were then aligned in MegaX. Datasets for all 13 protein coding genes were then concatenated in concatenating, with the most suitable model selected via J-model test. And the maximum likelihood phylogeny was then constructed in IQ-tree with 1,000 bootstrap replicates. On the resulting phylogenetic reconstruction produced using all 13 mitochondrial protein coding genes, there was full support for a monophyletic aculotta clade. Within the aculotta, there was full support for a monophyletic Siluridae clade and strong support for a monophyletic Palynuridae clade. Within the Palynuridae clade, there was no evidence for a Silenthes clade, likely because there are only complete mitochondrial genomes for 2 species. However, there was moderate support for a Stridentes clade, and within the Stridentes clade, there was full support for a monophyletic Penorus clade containing the fully supported sister lineages 1 and 2. I then tested for purifying selection via a pairwise comparison of Penorus echinatus and Penorus interruptus mitochondrial protein coding genes with the Penorus argus outgroup. Then the analysis produces an omega value that indicates the type of selection occurring. And again, omega is the ratio of Ka to Ks. So when omega is greater than 1, positive or diversifying selection is occurring. When omega is equal to 1, neutral selection is occurring. And when omega is less than 1, then negative or purifying selection is occurring. So the first selective pressure analysis found signatures of pervasive background purifying selection across all 13 mitochondrial protein coding genes, with all having omega values under 1. And this makes sense considering the importance of fully functional mitochondria for energy production and thus organism survival. I would, however, like to note that there was a slight relaxation in this purifying selection on a number of the complex 1 genes, or the NAD genes, and on both of the complex 5 genes, ATP6 and 8. Selective pressure was further tested among branches, across sites and branches, and among and on codons using the HiFi package in the WebServer data monkey. Selection was tested across the phylogenetic tree by assigning foreground branches that are predicted to undergo positive selection, and background branches, which are the remaining lineages not expected to be under positive selection. So two conditions were tested to test the effect of temperature on the NAD2, 4, and 6 genes in complex 1. I assigned Panorosis as foreground. And to test the effect of low oxygen levels on the ATP6 and 8 genes in complex 5, a separate set of analyses were conducted with Silaridae assigned as foreground. Hi5 was used to test 1 branch model, 2 branch site models, and 3 site models. The branch model, relax, identifies whether selection strength has relaxed or intensified. It compares a null model with 3 rate classes versus an alternative model that includes the selection intensity parameter K as an exponent of the inferred rate classes. The branch site model of Brezel is the preferred model for detecting positive selection at individual branches, and specifically it identifies the proportion of sites under positive selection. And this model compares a null with that does not allow for positive selection versus an alternative model that tests optimal rate classes, and using optimal rate classes acknowledges that different branches can potentially have different evolution pattern complexity. The branch site model busted identifies gene-wide positive selection on at least 1 site and at least 1 foreground branch, and compares a null model where omega is set to 1 versus an alternative model that has unrestricted rate classes. And it's important to note here that for busted, support is suggested via evidence ratios, which are log scale likelihood ratios that evaluate whether the alternative model fits best, but that evidence ratios should not be taken as statistical support. The site model mean identifies episodic positive selection on branch sites via a mixed effects maximum likelihood approach. 2 rate classes are inferred with weights corresponding to the probability that a site evolves under that class. Meanwhile, FUBAR is a Bayesian approach to infer constant Ka and Ks on sites and assumes constant selection pressure on sites across the tree. And FUBAR reports posterior probabilities on a scale with values greater than 0.9, strongly supporting positive selection. And lastly, SLAC uses maximum likelihood to infer the most likely ancestral sequences at nodes, and it uses counting to then infer Ka and Ks for sites. So this model also assumes constant selection pressure for each site across the tree. Overall, RELAX found potential signatures of intensifying purifying selection in Panoris, and potential signatures of relaxation and purifying selection in Silaridae. Brezel found potential signatures of episodic positive or diversifying selection in 8 of the Panoris protein-coding genes, namely in complexes 1, 3, and 4. And it found 2 potential signatures of episodic positive selection on 2 Silaridae protein-coding genes in complex 1 and 5. Busted found potential signatures of episodic positive or diversifying selection in Panoris complex 1, and no such signatures in the Silaridae. Mean found episodic positive selection for Panoris ranging from 1 site in Cobb, COX2, and NAD4 and NAD6 to 3 sites in COX1. For Silaridae, potential signatures of episodic positive selection ranged from 1 site in COX1 and NAD1 to 3 sites in NAD5. Both FUBAR and SLAC found no pervasive positive selection on any protein-coding gene. However, there was evidence for pervasive negative selection, which was lowest in ATP8 in both scenarios. So, to summarize, against a strong pervasive negative selection background, potential signatures of positive selection were found in almost all complexes encoded by mitochondrial protein-coding genes. The importance of these results depends on the function of the sites under positive selection, which these analyses do not reveal. However, similar potential signatures of positive selection have been found for other mitochondrial protein-coding genes in other invertebrates experiencing air temperature ranges, such as in hydrothermal vent crustaceans and bivalves, as well as cold water mayflies. And additionally, similar potential signatures of positive selection were also found in other groups experiencing hypoxia, including hydrothermal vent crustaceans, such as crabs, shrimps, and squat lobsters, deep sea corals, and deep sea corals, in addition to high altitude brine shrimp, fishes, birds, and mammals. The newly assembled mitogenomes of Penorus echinatus and Penorus interruptus represent new resources to aid with conservation and management via application in eDNA sampling and in species identification via barcoding. Pervasive negative or purifying selection suggests that the evolution of protein coding genes is primarily constrained to preserve the functionality of the OXPHOS pathway. And over the strong negative selection background observed for protein coding genes in the aculeata, potential signatures of positive selective pressure were detected within Panora species inhabiting tropical equatorial latitudes with consistently high temperatures, specifically in 9 out of the 13 protein coding genes in complexes 1, 3, and 4. And additionally, potential signatures of positive selective pressure were also detected in Siluridae species inhabiting depths of low oxygen levels, specifically in 6 of the 13 protein coding genes in complexes 1, 3, 4, and 5. So, to expand on the effect of temperature, adaptive evolution in Panora species likely preserves complex 1 functionality within the narrow temperature ranges of equatorial climates. However, as observed in a study conducted by Zhu et al. 2021 on the adaptive evolution of mitochondrial protein coding genes in mayflies that have an extensive cold water larval phase, positive selection in complex 1 is not exclusively linked to hot or cold conditions. Instead, it is likely that complex 1 protein coding genes adapt due to stress from prolonged exposure to narrow temperature ranges. So my study suggests that to preserve normal cellular activities in environments of narrow temperature ranges, positive selection on mitochondrial protein coding genes likely optimizes the functionality of all OXPHOS pathway complexes. Additionally, to expand on the effect of oxygen levels, adaptive evolution in deepwater Siluridae likely refines the ability of complex 5 to produce energy in low oxygen conditions. Across animals with disparate evolutionary origins, complex 5 protein coding genes likely adapted to low oxygen or hypoxic conditions inherent to the inhabited environment. My study suggests that mitochondrial protein coding genes in all OXPHOS pathway complexes likely undergo positive selection in low oxygen environments. Overall, these results are in line with other studies evaluating and finding adaptive evolution in mitochondrial protein coding genes in other invertebrates that endure narrow temperature ranges or hypoxia, such as in cave shrimps in a study conducted by Guo et al. 2018, as well as deep sea hydrothermal vent shrimps, crabs, and squat lobsters in studies conducted by Wang et al. 2017, as well as numerous studies conducted by Sun et al. And lastly, in similar signatures of adaptive evolution were also found in a recent study on deep sea corals conducted by Ramos et al. 2023. Despite evidence for positive selection of mitochondrial protein coding gene complexes affected by narrow temperature ranges and hypoxia, other environmental factors not described herein could also explain these adaptive evolution patterns in spiny and slipper lobsters. Additionally, many spiny and slipper lobsters also have ranges that extend into different climates or into both shallow and deep regions throughout different parts of their life cycle. This study evaluated adaptive evolution of mitochondrial protein coding genes based on nucleotide substitutions, but adaptive evolution can also occur via gene deletion, duplication, or differential expression. Although no gene deletions or duplications were detected, future studies should examine mitochondrial protein coding gene expression in detail. And lastly, species-level analyses are useful for identifying loci under selection, as I have done here. However, population-level selective pressure analyses are also needed to clarify patterns of genetic variation that are not well explained at the species level, such as the relationship between single nucleotide variants and environmental conditions. So overall, the study has demonstrated how plate tectonics and current patterns cause dispersal and bicurrence events that drive the diversification of Panora spiny lobsters, and specifically it highlights how dispersal events are likely more relevant to Panora's diversification than what was originally suggested by George. This study also demonstrates how environmental conditions affect the adaptive evolution of mitochondrial protein coding genes in spiny and slipper lobsters of the infraorder Acuata, and that potential signatures of positive selection are present in mitochondrial protein coding genes belonging to tropical equatorial Panura species and to deepwater Siluridae species. So thank you for attending my presentation, and at this time I would like to open the floor to any questions.